So, good afternoon, everybody. I'm Charlotte. It's really nice to be here this afternoon to talk to you. And I'm going to talk to you about predicting loop conformational ensembles. And I thought I should try and explain what I meant by that before I got any further. So, we're all quite used to the idea of we solve a protein structure and we get this shape. And let's imagine that I solved that nice blue protein structure there. Well, not me personally, I don't think that useful. But somebody did. And it had that red conformation for that loop there. Yep. And then if I was doing loop structure prediction, I'd try and predict that red loop. And I'd get it right or wrong. And we would be done. But let's imagine a few more people solved exactly that same sequence. And you won't be surprised to hear that happens quite often. And you discover that actually that red loop is sometimes where the red position is, sometimes where the white position is, and sometimes where the yellow position is. And that's clearly something that's really important, because it's those kinds of conformational changes that might be really important for understanding the function of the protein. So can we actually predict the different conformations of a loop, if it has any? And perhaps even more usefully, would we have any idea if a loop does have multiple conformations, or if actually it's static and it only has one? So what I'm going to do is predict loop structures using current methods. I'm going to use 4 different methods. You could pick your favorites. I suspect the results will be very similar because we deliberately picked a set of very different methods, and see if they can predict all the conformations of a loop. So that's the question we're asking. And the problem, as I set it up, was, of course, most of them are not used to doing this, though they're quite often used in this way. So when they're most commonly used in this way is when people want to think about docking or allostery or things like that. What people do is when they're predicting the conformation of the protein, they look at the 5 or 6 top predictions they got, and they say those are probably related to the various potential low energy structures of this protein, so they're telling me something about that structural variation. Well, the question really is, are they telling you anything about that structural variation? So we took the PDB. In fact, we just took everything in it that was better than 2 angstroms resolution crystal structures, and we found just over 5,500 unique sequences that had been solved more than once. So these are complete chains, been solved more than once, absolutely identical, and they contain 65,500 loops in that. And that's loops from 4 residues up. Okay, so 4 residues long loops upwards. And we collected what we called a loop ensemble. So that's a collection of structures for a given loop in this set. And there was on average 3.5 structures per ensemble. So sometimes we might have had 20 structures for something, other times you might only have a couple. The first thing I want to say, and I think this is quite important because everybody acts under the assumption that loops are going, or at least talks to me like that when I'm saying I'm doing loop structure prediction. If you look in the PDB and you ask the question, if something has been solved multiple times, is it moving? The answer to that question is no, on average. So only 1.6% of my loop ensembles have a variation greater than 2 angstroms. Now that doesn't mean they're not moving, because I can't prove a negative. It does mean the experimental data at the moment suggests that a lot of loops are not moving very much. And that shouldn't really be a surprise. Loops aren't actually sticking out from a protein surface. They're packed against it. They're part of the intrinsic structure of the protein. So the fact that some of them don't move shouldn't be a surprise. I was surprised that that few moved, but I'm still not surprised that most don't. And just to be clear that this isn't some really big length trick I'm pulling on you, and all the things that are short don't move and all the things that do, all the long stuff does, even if I go all the way up to 20 residue loops, and I still have quite a lot of data there, only under 4% of those have more than 2 angstrom variation. So you solve a 20 residue loop twice, it will probably be in the same place. That would be a very good bet, because 96% of the time you would be right. So taking that data, we created 2 test sets, a low variation set and a high variation set. So the low variation set, all the members of that loop ensemble are less than 0.3 angstroms apart. So basically, it's been solved multiple times and found in exactly the same shape. Now, I can't be definite that those things are low variation, just to be clear, because somebody could solve a structure tomorrow and show me it sits in a very different place. But I'm estimating that most of those don't move very much. And then I have a high variation set. And in there, I have at least 2 conformations that are more than 2 angstroms apart. So that's the 2 extreme ends of this distribution. And what I'm going to look at to show you data is 30 loop ensembles, and it's 5 for each length between 10 and 15 residues, because I've got a lot of data there, and it's probably fairer to look at the long loops than at the very short ones. So now a quick tutorial for those of you who don't work on loop structure prediction all day, every day. So kind of shaded out in the background there is my protein structure that I'm interested in. And then popping up in the corner there, you can see these things called N anchor and C anchor, those black bits pointing up. Loop structure prediction is very simple. What you do is you imagine you have your entire structure there, apart from the bit that's missing. So in this case, that would be that sequence, RGT. Yep. And all you're going to try and do is find possible conformations that can jump off from one of those anchor points and land on the other. And almost all methods work in a very similar way. They're slightly different around how they play with this. But what you try and do is a step called decoy generation. You generate lots of possible decoys that will go from one end to the other. You might do that by finding matching fragments in the PDB that will fit from one end to the other and are the same length as your thing of interest. Or you might do it by ab initio, if you like, connecting up those points. And you get this whole kind of stream of decoys that fit into that shape. And then usually you filter those to try and make sure they're not completely stupid for whatever reason. And then you score or rank them with your favorite energy function, statistical potential, pick what you like. So what I'm going to do in terms of this is I'm going to look both at the stage of decoy generation. Because I know what the answers are here, I can say, did I actually generate anything at all that looked something like the right answer? And I'll call that my best answer. So if I could select optimally from that set, I would pick that answer and be my best answer. And I'm also going to look at what I actually pick at the far end. So what did the method say was the correct answer? So on here, I've got an example set of results for a high variation target on the left and a low variation target on the right. So the first thing I want you to see, and you don't even really need to be able to read the numbers, is that on the right it's all blue, which basically means you got it right. And on the left, it's all red with a little bit of green, which means you got it wrong. And the first logical thing to think about here is kind of imagine what I'm doing. I'm actually making a prediction against every single one of those structures separately. Yep. I have lots of chances to hit the right answer if I'm in the high variation set, because every one of those is in a different place. So if you think of it like a dartboard, in low variation, I must hit the 20, because there's only one right answer. In high variation, I could hit the 20, the 15, the 11, the 3, it would still be right. So at some way, you would expect that I would do better with a high variation case, because randomly speaking, I've got more chance of getting it right. So if I'm just randomly picking, I would do better over there. But I do worse. The next thing that I'm showing on here, so the different conformations of the rows here, and then what I also have in the columns are the best. So the best possible is the most left-hand column we've got there. And you can see what happens is actually, even in the high variation, that tends to be green rather than red. So there's something sort of okay in my answers there, but I definitely don't pick it. So what you see here is I'm sort of worse at predicting them, but I'm also even worse at even getting them in that decoy distribution. So I've got these maybe thousands of decoys, and none of them look like these high variation targets. So to put this into a broader context, to show you I'm not cheating, because that was obviously a single case example, this is the average results across our entire set for 4 different methods. So the 2 methods at the bottom, Leap and Rosetta, are both ab initio methods. And the 2 methods at the top, so on the right at the top, Freed, is a knowledge-based method, and left at the top is a combination ab initio and knowledge-based method. And you can see in every single case, the pink bars are the high variation set, and the blue bars are the low variation set, that I am worse at doing the high variation. So it doesn't matter what method I pick, I'm worse in every single case. And to give you an idea of what it looks like, picture on the right-hand side here. So the native conformations are shown in the left-hand box. And for the low variation set, you can see all the native conformations sit on top of one another. And that's actually my top rank prediction, which looks remarkably like that set. I've obviously chosen a very good case example there to show you can do it. But the bottom is what tends to happen in high variation. So I have 2 different conformations, but the conformation I pick looks like neither of those 2. It's not even vaguely related. And I'm going to do a slight diversion, just to show you that we did think about some other issues. So the other way you could do this is to use molecular dynamics to do it, which is what I think most people would go to first. We did go to it first, but I kind of drop it in here because I find it less interesting in some ways. MD has exactly the same problem. It doesn't produce the experimental conformations. And it doesn't flip them from one to the other. So it doesn't even produce the MD minimized conformation of one of the other structures of a high variation set. So basically, what happens is that if you put in a high variation example, what tends to happen is it moves around a lot, but it doesn't go anywhere near any of the other high variation examples, which was very nice of it. And if you put in a low variation example, they all do roughly the same thing and move around less. So I suspect that MD might be very useful for telling you if something's going to be mobile, but it's not going to tell you what those shapes actually are. It tells you that there will be shapes, just not what those shapes are. So back to my sets. So what's the difference between them? Why can I do one and not the other? So the first thing is kind of quite a nice thing, because it's sort of obvious if you think about it. High variation loops tend to have smaller anchor separations. So if you imagine I've got a loop running between these 2 points, it's like a piece of string if it's 10 residues long. If I pull these far enough apart, there's only one possible conformation, because it'd be completely taut, only one way to get between them. If I move them closer together, there's loads of places I can go. And it turns out that's something you can see. So it looks like the high variation loops, maybe they have more conformational flexibility because they have this difference. The problem is, if we match the anchor differences, so if I recreate my sets and only allow things with the same anchor distances through, the prediction accuracy difference remains the same. So that's not what's causing our prediction problems a problem. It might be one of the reasons you can gain high variation, though. So the next thing, which is a difference between the two sets, is high variation loops tend to have a lower number of atoms of the protein chain they're on in their vicinity. So once again, this probably isn't very surprising, because in some sense you're saying they're less stuck down, they're less bound into the protein. Now, that might explain to us why we're unable to rank these structures, because of course I'll have less information to decide if this is a good conformation or not, because almost all the ranking methods are using some form of energy function to try and do that, which is usually based on contact to the protein. It's less explanatory of why you would fail to make the shape at all, because the shape must be possible, you just don't know how to get it there for some reason. The final difference is that more of the high variation than the low variation have atoms that are close that are not from that protein chain. So once again, you can think of this in the high variation. What you might be seeing is I've got an apose structure and then I've got the bound structure. And you go, well, yeah, then the loop moves. Except lots of times loops don't move when you do that, but sometimes they do. Now, we didn't do anything about that. We didn't ask that question before we started. We did what you would do in a normal modeling situation because you wouldn't have the partner. So none of the partners are there. So there's a kind of obvious thing here which is sitting over this, which is if I already had the crystal structure with the bound partner there, I'd probably get the loop in the right place. But if I already had the crystal structure with the bound partner there, I'd have the loop in the right place. So I have to perform the prediction without it there for it to be useful for someone to be able to bind the partner and do the docking. So this is a real problem in the sense that if this is what's causing the difference, then it's going to be very difficult to get around this. You're going to have to think about ways of dealing with that. So we divided the target sets into subgroups based on binding. What we found interesting about this is it is true, if I look at the low variation set, that when I'm trying to predict things which are bound, so in other words have an atom from a different protein chain near them, then I have more difficulty in getting those right. So my average RMSD is a lot higher than if I'm looking at unbound loops. So loops that don't move and are not bound to anything, you'll get right. What's slightly more worrying is in the high variation cases where I am not looking at binding, where you'd hope that the thing would drop and that would be the good cases. It's not true. So in the high variation cases, it's not just because of the fact that it's a bound and unbound structure that's making it difficult. It must actually be related to the kind of energy surface you're trying to predict against. So now I'm going to tell you some good news, because so far I've just told you you can't find the conformations of the loops you'd really like to see. So there is a little bit of good news, which is you can actually predict quite well if your loop is going to be high or low variation. So in other words, is it going to be mobile or not, just by running structure prediction. And what makes this interesting is I don't even need the structure of the loop to do this. Because it turns out that all I need to do is look at the RMSDs between all the decoys I've generated. So I'm not looking at the right answer at all here, I'm just looking at the RMSDs between the decoys I've generated. And the RMSDs between the decoys for the high variation, it's a much broader distribution. Whereas for the low variation, it tends to be quite tight. And once again, if you think about the problems I'm describing, that makes sense. When the program knows what it thinks the answer is, it keeps aiming at the 20, yeah? And so it hits it quite often, and you have a narrow distribution. When it doesn't know what it's aiming at, and apparently it really doesn't, you have this massive scatter, and so you've got much larger RMSDs between all your decoys. So, conclusions. I think the first thing I'd like to say, and I'd like to say it quite loudly, is that most loops do not have multiple different conformations according to the crystal structures in the PDB. So it's not true that loops don't move, but I think we need to be a lot less kind of, you know, everything's moving dynamically and throwing itself all over the place a lot. There are definitely large motions that happen, and they are definitely very important, but it is not clear that everything is in wildly different changing conformations all the time. Secondly, using current loop structure prediction methods, it's not possible to generate a set of decoys that reflect the diversity of the loop. And I sort of suspect it's related to a sort of very basic idea. If you imagine you have a low variation loop, what it probably has is a single deep energy well, because it's got one place to go, and that'll be relatively deep. If you have a high variation loop, even if it's talking about bound and unbound structures, what you're likely to have are multiple low, small energy wells. So none of them will be as deep. So what can we do next? Well, one of the first things we have to think about to improve this is actually testing methods to solve this problem. Because it could well be we've inadvertently trained our methods to be unable to do this because we've trained them so explicitly to solve the I want a single conformation and I just want that to be right, which may, for many questions, not be the right question to have asked. So I'm going to finish up by thanking all the people who helped me do this. So first of all, around the edges, all the money that allows me to do my job, which I very much enjoy. And then on the slide, in the picture in the middle, second in from the right-hand side is Claire, who did pretty much all the work I have just described. And then I have to finish with adverts. So obviously there's my own poster. There's a couple of posters from my group showcasing some of our work on antibodies, and a couple on our work in machine learning and drug discovery. And if you are interested in working with us on our antibodies and immunoinformatics work, please come and speak to me. Thank you.
Hi, Charlotte. I think you hit the right point there on the high variation set. I would say it's a classic example of frustration, and the algorithms try to at the same time satisfy opposing sets of favorable interactions that would take it one way or another, so they don't go anywhere. This is just a comment. My question to you, and we can discuss later. My question to you is that maybe you didn't have time, but you didn't quite convince me that loops don't move. I would ask you a few questions like, what about just NMR structures? What about pairs of structures of the same protein not solved by the same group?
So
all these, these sociological things around that.
So I think
we use,
yes.
We use the known structure in order to define
yeah.
the new structure.
So that's why that's why I was quite careful about how I phrased this. According to the data we have, it is very difficult to say that these things are moving a lot. And you hear people say all the time that they're moving a lot. And if they're here, if you look, the B factor is very good because we purposely picked very, very good structures. That's what we did. Because you can, that was the best control we could do. And we have plenty of examples where they're solved by different groups. I wouldn't argue that that means the numbers are as low as I'm saying on there, that only a couple of % of loops move. But I would argue that the numbers that do move they cannot be 100%. Because otherwise you're saying that every crystallographer is solving their structures very badly indeed. And I'm not brave enough to say that, so I think I'll... So we looked at that, no. So that was one of our things we checked on the list of differences between, but no.
So I was just wondering if you try this method on hydrogen bonded turns or beta bends, something other than alpha helix and beta sheet, so beyond just loops or coils, what would be
those would have been included in my definition of loops. So for me, a loop is anything that is not. Sorry, I should have been clearer. Anything that's not an alpha helix or a beta strand.
So how about we cluster them together in different classes? Would we see different results? Do you expect?
I suspect that you would, but I suspect that would be related to the fact that if you talk about the very specialized things like beta turns, they will be low variation loops. And I don't think anyone in the room would be surprised by that. And we would predict them very well also because they're very well understood class. So knowledge-based methods would just do it right.
Okay, thanks.
Thank you for the informative talk, and my question is kind of technical. So, can you comment on the possibility that for example, loops are moving, and they are moving so much that in the same crystal they have unresolved coordinates? So, can you comment on this part of possibility?
So that's sort of related to the question that was asked from the front about B factors. So, if you have very poor B factors, that would indicate that the crystallographer has been unable to, if you like, put the coordinates in a specific position. But more frequently, if they're really that unresolved, what you'll get is they don't put any coordinates in at all. So if you look at lots of crystal structures, the loops are just not there. Now, I won't see that here. Okay, so that's gone from my analysis. So it would be perfectly correct to say those are things that are potentially mobile. But I would argue in a sense that they are different type of mobile from what I'm talking about here. So here I am not talking about something that's moving very rapidly and could be in any conformation. I'm talking about something that potentially has a set of possible low energy conformations. So something that has change of conformation upon binding. So that must be a relatively low energy conformation for being able to take it up upon binding, or can exhibit two different shapes. So it's a slightly different type of loop.
Thank you.
Okay, let's stop this. Thank you.